You may have heard of hemoglobins before, and how they are made up of alpha chains and beta chains. But did you know that there are other globin chains out there that can make other types of hemoglobins? Let's take a look at some of these other hemoglobin variants and how the abundance of them may change throughout a person's lifetime. Hemoglobin is a protein that is found in our red blood cells. With its heme groups, it helps to hold on to oxygen so that this important gas can be circulated throughout our bodies. The type of hemoglobin that is most well known is adult hemoglobin, or HbA. And there is good reason for this because it is the most common type of hemoglobin in adults. But it hasn't always been the most common hemoglobin throughout all our lives. You see, hemoglobin A is the type of hemoglobin that is made up of 2 alpha globin chains and 2 beta globin chains, in addition to the heme groups that I mentioned earlier. But we haven't always made beta globin chains, and the different globins are produced at different times during development. We can see the relative levels of the globin chains being produced throughout early development by looking at this chart. The left axis shows the percentage of globin chains being produced, and the bottom axis shows the age of an individual from the moment they are conceived in months. We all start out with the production of alpha globin, and 3 other types of globin: gamma, epsilon, and zeta. As time goes on during the initial 3 to 6 months, the production of epsilon globin and zeta globin steadily decrease as their corresponding genes are repressed or turned off. In the meantime, some beta globin is starting to be produced as the gene for beta globin is starting to get activated. At around 9 months of development, shortly after we're born, an important switch occurs. The production of gamma globin decreases and is finally surpassed by the production of beta globin. This is when hemoglobin A finally becomes the dominant form of hemoglobin. With time, the production of gamma globin should decline until it is almost completely shut down. At the same time, the production of another globin chain called delta is being slowly ramped up. It will continue to be produced at this low level throughout our lives. Now that this chart is all filled out, you can start to see how creative the names are for the different globin chains. But more importantly, the next step is to figure out which of these globin chains can fit together nicely. Hemoglobins are examples of quaternary structure, usually made up of 2 pairs of globin, or 4 globin chains altogether. Like a puzzle, there are certain pieces that fit nicely with each other, such as a piece with a socket and one with a tab. To start, we know from the well-known hemoglobin A that alpha and beta chains should fit together nicely. Knowing this, we can then try to figure out which globins are more like alpha and which are more like beta. Zeta globin chains are biochemically more similar to alpha globin chains, hence why zeta and alpha globins are sometimes collectively referred to as alpha-like globins. Alpha globin chains are encoded by the HbA1 and HbA2 genes, while zeta globin chains are encoded by the HbZ gene. Interestingly, all of these genes are located on chromosome 16. The rest of the globins, including beta, delta, epsilon, and gamma, are sometimes collectively referred to as beta-like globins. Beta globin is encoded by the gene HBB. Delta globin is encoded by HBD. Epsilon is encoded by HBE1. And gamma globin is encoded by both HBG1 and HBG2. As you may have guessed, all of these genes are located on the same chromosome, chromosome 11. Going back to the chart, In the very beginning stages of life, the main globin chains being produced are alpha, epsilon, zeta, and gamma. These globins pair together to produce various forms of embryonic hemoglobin, with the three main types being hemoglobin Gower 1, comprised of 2 zeta chains and 2 epsilon chains, hemoglobin Gower 2, comprised of 2 alpha chains and 2 epsilon chains, and hemoglobin Portland, made of 2 zeta chains and 2 gamma chains. As weeks go by, production of epsilon and zeta slows, and the high amounts of alpha chains and gamma chains being produced results in the formation of fetal hemoglobin, or HbF, the main type of hemoglobin present in our red blood cells in the fetal stage of development. Both fetal hemoglobin and the various forms of embryonic hemoglobin actually have a really high affinity for oxygen, meaning they can hold on to oxygen very well. The greater ability of these types of hemoglobin to bind to oxygen helps in oxygen transfer from the adult hemoglobin in the maternal bloodstream to the fetal hemoglobin in the fetal bloodstream. Shortly after birth, hemoglobin switching from HbF to HbA occurs. This makes sense because alpha and beta globin chains are the most widely produced at this time. The adult type of hemoglobin doesn't hold on to oxygen as well as fetal hemoglobin can. But this is actually a good thing, so that the oxygen isn't stuck on the hemoglobin and it can hop off where it is needed in the tissues. The low levels of delta globin can also bind with alpha globin to make small amounts of hemoglobin A2, a minor form of adult hemoglobin that is present in adults. These are the main forms or variants of hemoglobin found in red blood cells throughout our development. However, there are other abnormal hemoglobins that can result from mutations to one of these globin chains. For example, sickle cell disease results from a single nucleotide substitution in the HBB gene. In this case, causing the replacement of a glutamic acid to a valine. This variation alters the resulting beta globin enough that it has different biochemical properties. The resulting hemoglobin from their binding with alpha globin chains is called sickle cell hemoglobin, or HbS. Although the alteration does not significantly affect the hemoglobin's oxygen affinity, it makes the hemoglobin sticky and clump together in red blood cells. This causes the red blood cell to take on a characteristic sickle shape and clump together in blood vessels, leading to pain crises and fatigue. Much like sickle cell disease, there are other mutations that can lead to alternate forms of hemoglobin, such as hemoglobin C, hemoglobin E, hemoglobin constant spring, and more.